Hello everyone. My name is Dr.Nathiya, Assistant Professor from the Department of Microbiology, Auxilium College, Vellore. Let us talk about plant microbe interactions. Plant microbe interactions. Plant microbe interactions are a kind of biological interactions between the species in a community have on one another. The mutualism is a positive interactions. The mutualism, each organism in the interaction gets benefited from the association. Mutualism is an obligatory relationship in which mutualist, that is endophytes, and host, that is plants, are metabolically dependent on each other. The examples are lichens, endophytes. Let us talk about endophytes in detail. The endophytes are the mutualistic symbiont harboring the living tissues of healthy plants. Endophytes are ubiquitous in nature, present in all the plants like bark, leaves, flowers, fruits, roots, seeds. The endophytes have a mutualistic association with the host plant so that produces the potential secondary metabolites. It can be a fungi, bacteria, or actinomycetes. The endophytes possess the exoenzymes necessary to colonize and grow well in the apoplastic washing fluid of the host. The long-time symbiotic relationship of endophytes with host plants have exchanged their genetic material. As a result of such mutualism, endophytes can produce the similar kind of metabolites as the host plants. The endophytes have a strong tolerance towards host unique metabolites or the toxic substances which produced during microbial diseases. Some novel bioactive secondary metabolites were produced during the detoxification process of endophytes. The endophytic secondary metabolites plays a vital role in various fields like pharmaceutics, agriculture, and environment, which supports the maintenance of human welfare. Let us talk about balanced antagonism hypothesis in plant. When endophytes invade host plant, the host defense mechanism gets activated and produces immune cells against endophytic virulence factors. Such produced immune cells limit the development of endophytes. At that time, the endophytes also produces the metabolites to overcome the host defense mechanism. If the endophytic virulence and the plant defense mechanism are balanced, the association between endophytes and their host plants will maintain a balanced antagonism. If the plant defense mechanism is defeated by the endophytic virulence factors, the association will turn into the negative interaction, that is plant pathogen relationship, and thus lead to plant disease. Let us talk about isolation of endophytes. The healthy and freshly collected plant parts like leaves, barks, roots, seeds, or the flowers will be washed with the running tap water and subjected to surface sterilization, initially with 75% ethanol for 3 minutes and then followed by 1% sodium hypochlorite solution for 2 minutes. Then the plant pots will be washed with the sterile distilled water for 1 minute, and this step is repeated for 3 times in order to remove all the sterilants added on it. And the plant parts will be cut into smaller pieces of about 5 into 5 mm. The samples were hand dried inside the laminar and airflow and place it over the required media and incubate the plates for 7 to 14 days for the colonization of endophytes, that is either bacteria, fungi, or actinomycetes, which can be used for further analysis. Let us talk about advantages of exploring endophytes. Endophytes are readily renewable. Imagine the 25 kg of Taxol production requires 30,000 of trees, 1 kg of paclitaxel requires 10,000 kg of barks, 1 gram of vincristine requires 500 kg of leaves. Instead, the endophytes can be produce the same amount of anti-cancer drug with a small amount of respected starter culture. The endophytes are ubiquitous in nature, present in all the plants, reproducible. It has a long shelf life, inexhaustible than the vulnerable plants. Critically endangered plant species can be protected, high yield and cost effective. To conclude, Endophytes can be produced biologically active agents on a large commercial scale as they are easily cultured in laboratory and fermented instead of harvesting plants and affecting the environmental biodiversity. Thank you.